So why don't we get started? It's a great pleasure and indeed an honor for me to visit Veldhoven. And I actually first came to the Netherlands as a graduate student myself. There was a series of summer schools on fundamental problems in statistical mechanics. And so I went to Wageningen, and it was the rainiest summer in 40 years, but otherwise it was very enjoyable and a lot of fun. It'd be great before I get started just to have a sense of my audience. I understand that most or all of you are working for the PhD. And can I have a show of hands, people that think they might go into some sort of aspect of theory? Anyone? Experiments? And people that might, although having been trained in physics perhaps, have an interest in biology? I presume some of you. Okay, good. I hope to get to know more of you maybe during the break and as we go along. What I often say when I give talks to any group is that it's a collaborative effort, and so I welcome and encourage interruptions and questions as we go along. And even though this is a distinguished group of students and so forth, I have a feeling that if you have a question, there are at least 5 other people around this table that have the same question. And if you ask it, they will be secretly grateful. And I will certainly be grateful to have your interruptions. And one of the reasons I enjoy coming to the Netherlands is it's a country where people don't hesitate to ask questions. So I relying on you to stop me. I'm going to be using PowerPoint. PowerPoint I think of as a form of asymmetrical warfare. I can just throw up all these slides and drown you in a sea of information, and your job is to slow me down. I will go to the whiteboard and do a few simple calculations which will also, I hope, slow me down, but I'm going to need your help. Okay, so we live in a remarkable time in human history because we, for the first time, can rapidly and inexpensively sequence DNA, not merely of humans, but of all sorts of organisms. And there was a milestone that allegedly was passed recently called the $1,000 genome. And this is a human genome, which can be at least approximately sequenced, maybe not $1,000, but relatively cheaply compared to the billions that were spent to sequence the first genomes of James Watson and other people back only 13 or 14 years ago. So this map comes out of that technological development. I won't go into the details, but it's the result of sequencing the mitochondrial DNA of present-day indigenous populations that have been around a long time, various parts of China, also in the New World, it's not shown here, India, and so forth. And by looking at those mitochondrial genomes, you can actually infer a migration history of human beings, a kind of family tree, but laid out on geography, laid out on a map of the world. And so one of the themes of this, of what I'll tell you about, is in a hopefully a context helpful for physicists, what happens when we have spatial population genetics, spatial migrations, and what's going on. If you look here, this is something we'll talk about in more detail. It's a population wave coming out of Africa. How many of you have heard about the out of Africa hypothesis for human origins? So it probably happened several times. It might be that the first Aboriginal Australians came out in an earlier wave, and then there were subsequent waves. We now can sequence Neanderthal DNA and see if humans had some sort of interactions with the Neanderthals. And one of the prominent migrations probably happened around 130,000 years ago. These are years before present. But there are two things that are a little worrisome about this beautiful picture. First of all, at least as I've presented it here in cartoon form, there are no error bars associated with the arrival times. Now, I can assure you there are error bars, and they're quite large. The second thing, regarded as an experiment, if we think of the context of say, physics or chemistry or biology or whatever, it's an experiment that's not easy to repeat. You probably wouldn't want to repeat it. And for this reason, we got interested, this is my colleague Oskar Halacek and I, in trying to examine the kind of questions that lead to these inferences in a Petri dish, in a dish that's maybe 8 to 10 centimeters across. And the idea is that whereas in 500 generations of, say, humans, or you could also apply it to cane toads invading invasive species coming into Australia, but for humans, at least in 500 generations, I'll try to convince you that there's a wave-like behavior that in 500 generations would enable this wave of humans to expand maybe 10,000 kilometers. And if you repeat that experiment, and I'll explain where this comes from on a much smaller scale, this is 100 microns here, in a bacterium called Pseudomonas aeruginosa, in the same 500 generations that produced this picture, that produced this idea of human migrations, you go a centimeter. And so the range expansions stay in the Petri dish. And you can do it 40 times and try to understand these patterns and try to understand where they come from. So if you don't mind a change in length scale of 10 to the 9, we can perhaps begin to understand some aspects of spatial evolution in a simple, reproducible context. So with that introduction, let me go back. This is the abstract of what I was hoping to cover. And I won't have time to cover everything in detail. And I'm not planning to. So there's plenty of time for questions. But if you want to know more about some of the math or some of the ideas, there's a popular article that appeared in Physics Today a couple years ago. And then there's a more detailed review article in Reviews of Modern Physics where we discuss these ideas of genetic demixing and evolution in something called the one-dimensional stepping stone model, which is a famous model in population genetics. So any questions so far about where we're going? Have I made myself intelligible? Okay, so in this context, if I look at bacteria growing across a Petri dish, and if I have time in the second half, I might show you a movie, what's going on? So what we have here is a picture of a monolayer of bacteria pushing their way upwards. And I want you to imagine that the left half is a species which is very similar to the right half, but it has a marker on it. We'll talk more about this later. It's a fluorescent marker which under the right excitation shows up red. And on the left side there's a marker that shows up green. And so the idea is that there only maybe the first 20 layers of bacteria. These rod-shaped organisms are E. coli. And can anybody hazard a guess on the number of E. coli in a typical adult intestine? Anybody know? Wild guesses are allowed. 10? A billion. Okay, thank you. There are more E. coli in your intestines and mine as well than there are people on the surface of the Earth. And you get that by just calculating the volume. The length of this rod is 2 microns, micron across. Think of it as a cylinder divided into the volume of a typical person's large intestine, which is where they live. And you'll get 10 billion, 12 billion. So there are a lot of them. Not so many on this Petri dish. And the first 20 layers or so are the ones that actively divide. So there's a kind of range expansion that's happening due to cell divisions in a relatively thin layer at the frontier of this population, leaving behind perhaps a mixture of red and green, and that will be one of the questions that we're going to try to address. Is what is that mixture? What does it tell us about the range expansion itself? And there's all sorts of interesting scenarios, some of which we'll try to understand today. For example, the red and green strains could be neutral. They could be genetically identical in almost every respect except for their color. These are typically grown in a dark refrigerator, and so they can't tell the color. But what I want to ultimately convince you of is that there's a boundary, kind of wiggly boundary between red and green, as illustrated here, which behaves like a random walk. Okay, how many of you have studied random walks in your various courses on statistical mechanics or polymer physics or whatever? So why is it a random walk? Well, let's imagine I have one generation of growth, so I go out another 10 bacterial thicknesses. And if the green accidentally reproduces the red during that generation, this walk will bend a little bit to the left. But if red were to do the same, it would bend a little bit to the right. And if they're selectively neutral, the discrete number fluctuations at this interface, at this frontier, will cause this thing to wiggle back and forth. And the statistics of this wiggling boundary, this genetic boundary between red and green, behaves like very much like a random walk, where this direction is like time, because as I'll try to show you, these populations typically advance at a constant velocity. So distance is a little bit like time if you multiply by velocity, and then the transverse wiggling is like a one-dimensional random walk in the perpendicular direction. We might call that x. Yes, please.
Why do they only multiply in the top region?
Okay, great question. Typically what happens is they're sucking up nutrients. They're sitting on an agar Petri dish, so it's like a jello. In this jello are lots of nutrients, and by the time this population goes by, the bacteria back here have sucked up virtually all the nutrient that's available. They don't die, but they reprogram themselves to stop cell division. Thank you. Any other questions? Yeah, please, there.
But that would be pretty different from humans, right?
Yes and no. Good question. We humans don't go into stasis as soon as the population goes by. That's a good point. On the other hand, what happens for humans, and you can do this with bacteria that swim in soft agar, is that you swim around aimlessly in a two-dimensional random walk, and this boundary will slowly diffuse away, but because the frontier is advancing linearly in time, you develop long-lived sectors, sectors which can persist to the present day, which are only blurred out by a much slower process, which is this two-dimensional diffusion that you bring up. You can do experiments that mimic that, but we think that even for humans, which are diffusing, the sector structure that I'll show you and that I showed you earlier, for example here, is remarkably long-lived. It may blur out back in the wake, and eventually all the signal will be lost, but it's a very slow process. There was a question there. Anyone else? Yes, please.
What are the initial conditions of that experiment? I mean, they were in maybe the same place?
Okay, yeah, so I'll tell you in more detail, but I can tell you briefly now. If I go back here, this central region here, we like to call the homeland. And so what we did was, or what this group of experimenters did, this is Kevin Foster and colleagues, was to grow up the red and the green strains overnight in a liquid culture, shaking them vigorously, mixing them up and so forth. And then in the morning, and I've done this too, you take a small portion of the mixture, say a 50-50 mixture, or it could be some other proportion, in a carrier fluid, and then you pipette it down, maybe 5 microliters, in the center of a Petri dish. The carrier fluid dries out in about 3 minutes, and then you have an initial condition that you asked about of fairly well-spaced bacteria. There's an excess of them on the rim. If we have time, we can talk about that. And then the expansion starts to take off. The dividing time of these bacteria is about once every 36 minutes. So this maybe took 4 days to grow up like this. Other questions? Okay, so we have this random walk boundary, perhaps, but if the green is out reproducing the red, we would then say there's a selective advantage. The green breeds faster or produces daughters faster. Bacteria are simple because they have haploid asexual reproduction. Mothers divide and their daughters divide, so it's kind of like an eternal process of simple cell division. And if that's the case, then this random walk would systematically bend to the left if the green is out reproducing the red. So it would be a biased random walk. And it could, of course, go the other way. They can also be kind of nasty, these bacterial strains. One or both could secrete toxins that impede the other. And some bacteria do that to foster their own growth. And finally, and I hope we'll get to this, you can have mutualists. Mutualists are bacteria or yeast or some other microorganism that they force them to play together because one or both could secrete amino acids useful to the other. And so then they actually don't like to be segregated. I'll give you an example later for yeast where, say, the patch of organisms on the right can make leucine but not tryptophan. With the reverse on the left, they can make tryptophan but not leucine. So these are two strains that need to get together and be well mixed. And yet if there's a tendency, and I hope to convince you that there is such a tendency for this genetic demixing, that sort of fights against cooperation. Not inevitably torpedoing cooperation, but it's another interesting issue, and you can create strains in the lab that do all these things. Other questions? Okay, so this is what I'm going to tell you about. We're going to focus on frontiers. Why frontiers? Range expansions are very common in biology, not just humans. And at the frontier, where the population is very sparse and thin, those number fluctuations are going to play an extremely important role. And you'll see that it affects population genetics, and it can bias survival of the fittest, which we all learn about from Darwin, into survival of the luckiest. And both things are going on, both are important, and it's very much like trying to understand random walk with a little bit of drift, a little bit of bias. And so these biased random walks will be going on a great deal in this lecture. And we're going to, as I said, try to see if we can test theories of frontier evolution and cooperation with colored bacterial strains or yeast strains with variable mutualism. And hopefully, I'm pretty sure I'll be able to do this, I'll get to a famous model, sort of like an Ising model or an Anderson model or a Hubbard model in condensed matter physics, which play a prominent role in posing and challenging questions. There's a stepping stone model of cooperation and competition that I will tell you about as part of this lecture, which underpins much of spatial population genetics, and we can actually do kind of a spatial version of game theory, as you'll see, with these organisms, as you might have guessed when I talked about poisoning and mutualism. And you can even get some phase transitions, speaking of the Ising model, in these range expansions that might even be important biologically. Of course, this is not work that was done by myself. It started with Oskar Hlacek, and I've had enormous good fortune in being able to work with a whole bunch of very talented people whose work will come into play as we come along. So here's, again, one of these range expansions, just to focus you. This is different colors. It's blue and yellow, I think. This is a movie of a range expansion of Pseudomonas aeruginosa. And it almost looks like oil and water phase separating, okay? But it's not. In fact, these two strains, they're colored versions of this picture here of the Pseudomonas aeruginosa organism, are as identical as we could make them. They differ only in the color that is expressed, and that only under fluorescent excitation. And they're grown in the dark. And yet, even though they're quite similar, they're not chemically different, the way oil and water are chemically different. They phase separate, but it's not like chemical phase separation. It's like if you leave salad dressing in the refrigerator for a year, as I've occasionally done, you find a lighter fluid at the top and a heavier fluid at the bottom. It phase separates. What's happening here is the descendants of this inoculation around the homeland is what's phase separating. Pseudomonas aeruginosa is an interesting organism. It's a pathogen. You don't have to worry too much about it working in the lab if you have a good immune system. But it goes after people with cystic fibrosis. And so there's a lot of interest in trying to impede its range expansion. So here's a simulation which could be a range expansion of two different colors along a catheter tube in a hospital that unfortunately wasn't taking adequate precautions to keep these bacteria outside the hospital room. And so it's worth understanding how they migrate, what's going on with the genetic demixing. And one of the things we'll see is there's two kinds of waves that play an important role. One is a population wave. So if I just looked at this in white light, you couldn't tell that there were different genetic variants segregating out, genetically demixing. There would be an outward velocity of this wave. I'll show you where that comes from in about one slide. So that's what's called a Fisher population wave. This is like the wave out of Africa. But here, along the frontier of this circular wave, we can see there's genetic variation, and there's a boundary between yellow and blue. And so at right angles, there's the potential to this population wave. There's a potential for a Fisher genetic wave where yellow systematically squeezes out blue. And if there's time in the second half, I'll show you why that might be relevant to simple models of the tumors that give rise to cancer. I hope we'll do that in the second part. In any event, there's a genetic wave in principle at right angles going along the rim here. We'll come to that, I hope. And here, because they're neutral, that genetic wave is sort of stalled out. This is just some sort of random walk. It's not really going anywhere except for random fluctuations. Those fluctuations, notice, can cause this yellow genetic domain to pinch off as it gets engulfed by the blue. But otherwise, there's no systematic motion. But if the yellow could out-reproduce the blue, you might imagine that there would be a perpendicular genetic wave going at right angles to this population wave. And of course, I have to tell you what waves are. You know, it's not like sea waves. It's a wave of a type, but it's a very unusual kind that is unfamiliar to most physicists. So these waves arise in the following way. This is a one-slide summary or two-slide summary of all of population dynamics. There's no genetics yet, just all population dynamics. And so the ingredients, the initial ingredients, this is has a illustrious history. If C is the number of organisms in a test tube, these little B's are bacteria. And if you put them in a rich nutrient broth and they grow up overnight, you'll have many more of them the next morning. And if I just focus on births and deaths, there's a famous calculation that goes back to Malthus in 1789. Malthus's book on the population, sometimes called the population bomb, erroneously, it's not really a bomb, but what he said was that the time rate of change of the number of individuals, could be this test tube, could be humans on the planet or whatever, is going to increase proportional to the number that are already there. And because they're having offspring proportional to how many that are already there. And so if births exceed deaths, this constant A is positive, and this would lead to exponential growth. And an exponential is not an explosion per se, but it's a pretty fast growth. And he was extremely worried about that. Darwin read Malthus and was quite aware of this exponential growth, and it certainly figured in his ideas about evolution. I don't know why it took so long, but Verhulst, who I believe was a Belgian scientist, recognized that this couldn't go on forever, and that competition for nutrients between organisms, even in a well-mixed test tube, would have to limit the growth. And so he said that there's going to be a negative contribution proportional to the density squared, basically, of these organisms. And so all the terrible things associated with, I like to say, death, famine, destruction, the four horsemen of the apocalypse, are summarized in the coefficient B. So that's a nonlinear differential equation, a very simple one. And you can solve it and compare it against the biomass in a well-stirred test tube of yeast. And what does it say? It says it rises up exponentially, but then begins to feel this coefficient B, bends over, and then saturates out at this fixed point value, C star, that's given by A over B. Okay, so that's the Verhulst picture of a well-mixed population. Now, humans, as we saw, involve spread often by spatial migrations. And they're not in a well-mixed test tube, except during the present time when everybody can hop on an airplane, and I could jump on a plane in Boston and get over here fairly easily, and vice versa. So now diffusion is sort of really, really strong, and it's almost like the world is becoming, in this era of globalization, a well-mixed test tube. And if you fit the exponential growth of human populations to this Malthus-Verhulst theory, you and we are here. So at about the year 2000, we passed the inflection point. And you can decide whether that's a good or bad thing.
And what would be the time scale? 20 hours?
Yeah, so this is for yeast. And indeed, these are hours. And so in a typical test tube, the yeast would grow up to this inflection point in about 8 hours. Their doubling time is about 90 minutes. Humans, different time scale, and I don't know, hundreds of thousands of years, but we are approximately here. But before we became well mixed by virtue of efficient transportation, how did organisms get around? Well, the simplest model was written down actually 100 years or so after Verhulst by R.A. Fisher, famous British population geneticist. And he said, well, there's a population density, say a two-dimensional density in space, could be on a Petri dish and so forth. And so in addition to the Malthus and Verhulst terms, he just said, well, there's spatial diffusion. So these could be humans aimlessly wandering around, or bacteria with flagella, or anything that's just floating. It could be ordinary Stokes-Einstein diffusion due to Brownian motion for small enough organisms in a test tube. So there's a diffusion constant D. And what Fisher and independently Kolmogorov and collaborators were able to show in that case in the former Soviet Union was a very interesting characteristic of this equation. It's a parabolic equation. It's not, when linearized, a wave equation at all. It just has growth, saturation, and diffusion. But it turns out this nonlinear partial differential equation admits wave-like solutions of the form of function x minus vt. And you can put this in and differentiate and show what equation this function f satisfies. I encourage you to do that. And the hard part is determining this velocity. But it's sort of plausible in hindsight. What happens in the early stages, if I say in one dimension, just as a one-dimensional Petri dish, let's say, or a little strip on a Petri dish, is the thing locally, if inoculated at the origin, grows up exponentially due to this term. And then, due to this linear diffusion, there'll be a spread. And so you probably know that diffusive processes spread, controlled by this diffusion constant, like the square root of time. But that only happens until C gets large enough to reach this ceiling in the allowed value. And eventually, this wave, this growth, hits the fixed point value. And so here, the population is saturated at what's called the carrying capacity. And then, sure enough, there's a constant velocity outward motion on the right and a similar motion on the left here, where that velocity is given by the geometric mean of the diffusion constant times the growth rate. And up to the factor of 2, which is the hard part, you can estimate that by just looking down here in the foot of the wave when the linearized approximation works and say, okay, I grow up at a rate A and I spill outwards due to diffusion at this rate. If you put those two things together, you can convince yourself that it's at least plausible there's a constant velocity. And there's also an interfacial width. If I beat this term here against this linear term, there's a characteristic width which is given by the square root of this ratio of the diffusion constant, which has units of centimeters squared per second, and this growth rate A. Okay? Yes, please.
Are you aware of certain conditions that parabolic equations should have to admit such wave-like solutions?
I don't know all the details. The mathematicians have indeed tried to look carefully into this, and Kolmogorov and his colleagues were great mathematicians. I don't know if they, in the 1937 or whatever it was, that they proved it rigorously, but we certainly know that this equation, and if you can change the nonlinearity a little bit, for example, still has wave-like solutions. And certainly on a computer simulation, you can robustly show that's what happens eventually. Notice this is two waves, one off to the right, one off to the left, not an isolated wave. And as I said, the subtle thing is the so-called velocity selection problem, which says that when you solve it, that the actual coefficient is 2.
Does it also depend on the boundary conditions that you use?
Well, in general, yes, because if I had walls of a Petri dish over here, the wave would eventually collide with the wall and so forth. But in a large enough Petri dish, before you get near walls, this seems to be the stable form that this nonlinear parabolic differential equation achieves. Other questions? Okay, so that's a wave, and I now want to give you approximately one slide, but then I'll go to the board. Summary of all of population genetics. We're just doing population dynamics. Now let's talk about genetics. And let's talk about it in a very simple context where this circle here is what would happen if you could look down on a test tube that had bacterial organisms of two different types, little a and big A. So presumably all of you, how many of you have studied in some stage in school, primary school, whatever, high school, the population genetics of blue eyes and brown eyes? Big B, little b. Okay, so these are haploid, so there's not two chromosomes, there's just one, and so it's big A and little a. It's really simple. It's simpler than blue eyes and brown eyes. And what this is intended this cartoon is intended to illustrate is that you could imagine an experiment where you start with only 8 of these organisms. And let's suppose 4 of them are capital A and 4 of them are little a. And let's suppose they have labels on them so you can keep track of the descendants. But otherwise, for the moment, there's no particular bias or selective advantage. But you let them grow up in the test tube overnight, and they're doubling every 36 hours. So after a full day, they may have doubled 40 times. 2 to the 40th is a big number, but maybe they're hitting that Malthus-Verhulst ceiling. You have a very full test tube, and here the test tube has elastic walls, and it's so full it expands in this little cartoon. So the experiment, which actually mimics in many respects the evolution of real bacterial populations, is to pipette the same amount, as close as you can get, that you had here out of this potential set of next generation bacteria and put it back in the test tube. Same fresh medium that you had here and the same number, just to keep it simple. And doesn't have to, of course, end up being 4 capital A's and 4 little a's. If this number n, which is 8 in this example, is reasonably small, there'll be binomial fluctuations. Random chance might in the next generation produce three capital A's and 5 little a's. And if I keep going for a very long time, and I start here with, let's say, a population with 20, and I let these things sort of random walk around, I start off, let's say, with 1 capital A, and I follow this procedure over and over again, which I can easily do on a computer. What you get looks very much like a random walk. Okay? It jumps around. However, it's a very special kind of random walk, not often encountered in most areas of physics. One reason why it's special, but this isn't the most important, is that there's absorbing boundary conditions. So if I neglect infrequent mutations, and I just ask what is the fate of 1 capital A, it will often go extinct, especially if it starts off with only 1 copy of itself. You might have 2 the next generation, and then it'll drop down to 1, and then to 0, and so forth. But occasionally, it'll get lucky and go all the way across in this is allele frequency space. Now, I should say, you all know something about biology, so I don't have to probably tell you that frequency is not measured in hertz. Frequency is the fraction of the population. So that's what I mean by frequency. And so it can go from 0% to 100%, and there'll be this perhaps one lucky strain or lineage which manages to random walk its way across this chasm. And it's a random walk with absorbing boundary conditions. So if it ends up down here, it never gets off this axis within this approximation. If the A takes over and goes from 1 out of 20 to 20 out of 20, it also will in fact stay forever on this upper boundary or this lower boundary. So it's a random walk with absorbing boundary conditions. And what a great population genesis of the 1950s and early 60s showed, Mutu Kumura, was that just like in physics, if this were a conventional random walk with Brownian motion, we would describe things in terms of a Fokker-Planck equation. There's an equation for the probability distribution here as well. So U is the probability that allele A has a certain frequency, a certain percentage of the population at time t. N is some fixed number, that the number is maintained at the same value, just to make life simple. And you end up with this strange looking diffusion equation that describes what's going on. And this diffusion constant depends on the allele percentage or frequency, so P is like percentage, divided by the population size. And so it's just binomial statistics. So there's a genetic diffusion constant that comes inside these derivatives that controls what's going on. And this might be a good time to let me show you, if I can, a little computer program which you can download. It's free on the web, called Populus. Have any of you played with Populus? So I'll show you how it works here. We're going to go down to Mendelian genetics. We're going to go to genetic drift and run one of these things. So what is this program doing? It's just what I described. In fact, the picture was taken from this Populus program. So this runs, so there are population size is 8 instead of 20. It runs 3 n generations. That turns out to be about how long you need to have most of the random walks annihilate or get stuck at 100% or 0%. And it says here number of loci, that's population genetic speech for the number of sites on a genome. Each site on a genome could independently be going on these random walks. Each nucleotide choice could be doing this, for example. Not always, but sometimes. Here they're starting off at 50%. And in analogy, so how many of you have studied Brownian motion? How many of you studied Langevin equations? Fokker-Planck? So this will be somewhat similar. Starting off at 50%, you could end up here or down there. And you can play around with these numbers, for example. I could make it 8 different realizations, if you want, of this random walk. And I could start it off at an initial frequency. Let's maybe go up to 20 here, just for the case that we were talking about. And so I'll go down to initial frequency of 1 individual. So if it's 1 out of 20, what should the initial frequency be? What's the smallest initial frequency that I could have that's not 0? Be 1 out of 20, otherwise known as 5%. Okay, so let's see what happens when I do that. I'll put in a 0 there, and I will run this thing. Okay, well, good, they all fixed, whatever that means. Okay, so look, here we have the danger of the absorbing boundary conditions. This is our neutral model, started off down here, and sadly, all of these realizations by generation 21 hit the absorbing boundary condition, and in every case, the allele, capital A, which was only 1 out of 20 to begin with, went extinct. It's a very sad outcome. So this is a little more fortunate, because this lucky black allele managed to random walk its way to fixation. Okay, so this is, and again, it's important that n is small. So Iceland, the Icelandic population started off with a very few number of founding members, and the number was relatively small, not 8 or whatever, or 20, but there's often there's a genetic level of genetic purity, because many possible genes or alleles have fixed. Allele is fancy population genetic talk for variations like red and blue, like brown and blue eyes and so forth. So here's one that actually fixed, although these other ones sadly went extinct. And you can play this for hours and amuse yourself in various ways. Okay, so that's what we want to study, and we'd like to study that with an aim to determining how a probability distribution evolves, starting with some initial condition as a function of time.
Question?
Yes, please.
So where does this sampling occur in nature?
Well, that's a great question. So one simple example. So there's certainly, you get very similar kinds of math by some sort of random mating model. So if you have an organism with 2 chromosomes like humans, and they mate promiscuously, then they get some random pairings, and a very similar kind of analysis as Fisher and Sewell Wright showed will give you a kind of a random walk. Sea urchins in the ocean spew out their eggs and sperm, and it literally is random mating. And now for these bacteria at the frontier, what's happening is not survival of the fittest. That's what I wrote at the top, but it is indeed like this random situation that I described, and it's survival of the luckiest. And the reason that happens is if I have one of these frontiers, and let's suppose I have a solid and a open circles representing my 2 types of bacteria, they're really rod-shaped and so forth. But what happens is in a very simple model where I just have one layer, there's lots of stuff down here as well, the wave, the Fisher wave is coming up here. What can happen in the next generation is either I could have a mother with an offspring of a daughter, if she's quick enough, that goes here, thus blocking this one, or this could do the same to its neighbor. And so even in this very, very tiny population size, like 2, effective population size is about 2, you can have these fluctuations. What happens in a more realistic model would be that everything going back maybe 10 layers of bacteria will be dividing, but no further. And then it's like a crowd going forth, jostling forward another 10 layers.
So the essence is the small size of the...
it's the small size of the two small sizes. One is in the third dimension, it's just typically one cell thick in the bacteria, so that's a small size, and then only 10 this way. And so roughly speaking, you could say that the effective population size is maybe 10 to 20 that are jostling forward in the next generation. So that's where it comes from in the microorganisms, and there's similar effects elsewhere. Yes, please.
I don't really understand then. It's still a bit of fitness too, right? I mean, if the white cells always are a bit quicker than the other one.
Absolutely. And so right now, just to make it simple, I'd like to focus on a situation where they have exactly the same fitness, so exactly the same cell division time, both 36 minutes. But then we, of course, have to look at what happens when I have a selective advantage superimposed on these number fluctuations. And to jump ahead a little bit, what we'll see is that instead of having a kind of neutral random walk with the same probability of going up as down, it'll become a biased random walk. And so you'll be helped on your way across this this dangerous region between 0% and 100% by some survival of the fittest. There'll be survival of the fittest interacting with survival of the luckiest. Okay. Other questions? Okay, so I'll just mention on a technical aside, this is like a Fokker-Planck equation. There is an analog for population biology of a Langevin equation, and for this simple problem of it's called neutral genetic drift. And drift is used in population genetics to mean unbiased motion. It's the random walk aspect of it, even though in solid state physics we think of a drift velocity of an electron or something in a metal. That's not what I mean by drift. But this random process, which sometimes is called drift, with this genetic diffusion constant, ends up in a Langevin equation. You just get that dp dt is some Langevin noise source with delta function correlations, but now this nonlinear prefactor comes out in front. And a subtle thing which you can read about in those references I gave you is that to interpret this stochastic differential equation correctly, in other words, to get this differential equation, which I'll show you how to derive in a minute, you have to use not the fashionable physics interpretation of stochastic differential equations. After all, it's nonlinear, so it's going to be subtle even on a computer. How do you step this forward in time? You have to use something called the Ito calculus. And it arises because in population genetics and evolution, the previous generation is what determines the statistics of the next one. And the continuous time Stotanovich interpretation, for if any of you care and know about this in the audience, is not the right one to use here. But if you're uncomfortable, this has a rock solid derivation, and this is just a way of getting a shorthand in stochastic differential equations that will give rise to this one here. Okay. So I'm now going to at least sketch the derivation of this equation, so you have an idea of what I'm going to be talking about. And just to remind myself, I guess at 9:15 we should stop and have a break for coffee. So if you notice me rambling onwards, you might want to just raise your hand and say, it's 9:15, and I will stop, and we'll come back at 9. So anyway, what's going on? How would you derive this kind of thing? Well, I'll just sketch a derivation that is kind of intuitive, and it actually would work for bacteria that are being jostled around in what's called a chemostat. So this is a test tube, but I haven't shown the extra connections that make it a chemostat. Who has played with chemostats? Anybody around the table? Anybody know what one is? So it's like an incubator, and all it's missing, because here's a bunch of cells, right, red and green, jostling around, and what it does is fresh nutrients are constantly fed in. So rather than the serial dilution that I showed you, fresh nutrients come in from the left, and waste material is taken out on the right, as well as dead cells, And this test tube is always at that Malthus-Verhulst equilibrium fixed point value, up to fluctuations. Okay? And to make it simple, let's just assume there's always a fixed number n inside the cell. And the model that I'm just going to very briefly sketch for you is called the Moran model, and it's just makes the calculations very, very straightforward and easy and intuitive. And then this Moran model turns out to give the same results as more sophisticated models. There's a kind of universality in the long time, large but finite number limit. Okay? And one of the themes of a lot of what I'll say is there's an order of limits question in population genetics. We'll typically be working in a limit where the number of particles is big but not infinite. 20 is pretty big for some purposes. And in a limit where there's a selective advantage. And we'll define this in a minute, but it's a selective advantage is a dimensionless number that describes how much faster the reproduction is. So if I had a bacterium that would normally reproduce in 100 minutes, but it has a more vigorous neighbor, it only takes 98 minutes to reproduce, then you would say there's a selective advantage of 2%. So this is typically much, much less than 1. And so we're going to eventually see that it's the product of n times s that determines things and determines the importance of genetic number fluctuations versus the bias, and survival of the luckiest is measured by this product of a large number times a small number, and of course that could be anything. So that's sort of where we're headed. Okay, so this model, the Moran model, came 50 years after the original insights into population genetics. It's just a lot easier to calculate with. It's sort of set up for the chemostat in mind. And what do? You randomly select 2 cells from this test tube, or this chemostat, one of which is allowed to reproduce. That will typically happen. And the other will die because there's only enough nutrient for a certain fixed number. And if you pick them, if they're neutral, and there's a fraction f that are green, then this event where the green reproduces and effectively one green replaces a red occurs with probability f times 1 minus f. Okay? Of course, it could go the other way. You could pick out the red one first and say, okay, you reproduce, but the green, you pick out a green, and that's the one that dies. And so now, instead of increasing the number of red cells by 1, you decrease instead of increasing the number of green cells by 1, you decrease the number of green cells in favor of a red cell. And of course, you could also pick out accidentally a green and a green. And in that case, nothing happens. That occurs with probability f squared. This occurred with probability 1 minus f to get the first red, and then probability f to get the next green. And of course, you could pick 2 reds, which occurs with probability 1 minus f squared. Okay, so that's what we're going to be studying and thinking about, this very, very simple model. And basically, you can think about it by drawing a kind of ladder of states. Okay, so this would be time. And one of these Moran steps, to really have a step of real time, I'm only acting on one cell or one pair of cells, and of course they're all really competing and dividing in parallel. So in fact, if delta t is the time step that I showed on this slide, I have to do it n times to let the whole population have the option of reproducing itself or dying. And so the generation time, the 36 minutes or whatever, in this Moran model will be capital N, that's the population size, times this delta t. Okay, so over here I have time measured, say, in generation times. And it's sort of, it's overlapping generations in the sense that you sweep through. I could do this on a computer, the whole population, and one by one you pick them at random. You advance at a unit tau g, and then there's also discretization in the vertical axis, which will be the fraction f, and that'll go from 0 to 1. All right. So what's the spacing of these discrete quanta of f on the left axis? Francesca, you might know. 1 over n. Because what happens, thank you, is that a particular value of f will either go up on this time step delta t to, you get one more of these guys, so f goes from what it was to f plus 1 over n. That happens with probability f times 1 minus f. It could stay the same. That happens with probability, that's the f squared possibility that I showed at the bottom. And then there's a 1 minus f squared possibility. And then it could go down, as Francesca said, to f minus 1 over n. And that happens with probability 1 minus f to pick the red one and f to get the green one. So what we have is basically transitions going up and down this ladder. And this is a job for the master equation. This is going to define how the probability evolves forward. And so what really is happening in this little picture is that I might be starting here. So I start with some value of f, say f naught, and then to get to the next generation, suppose this were delta t. I'm going to repeat it over and over again, but that first step, all that can happen within this Moran model is I either stay the same or I go up one rung of the ladder or down one rung of the ladder with these probabilities shown over here. And when I say this is a job for the master equation, I simply mean that this jiggling that is going to happen can be described by, so let's say the probability is u of f. We want to know what is the probability. So if I repeat this process, you can see that I'm basically going to be getting some kind of, basically to figure out what the probability distribution is out here, I have to sum over paths. Basically, I'm doing a path integral. It's like a Feynman path integral, but it's not for a wave function. It's for a probability distribution. And so if I ask myself, what is the probability of having frequency f, given that I started at frequency f0 at time t equals 0, how do I figure out what happens in the next generation? Well, I want to know what happens delta t later. Okay? And this I can write in terms of these transition rates. Okay, these transition probabilities. And so what I'm going to do is call these transition probabilities, I'm going to give them a name. So the name will be some rate phi. And these rate phi's are going to tell me what happens to f in one of these delta t Moran time steps. And the three forms of the function, namely f times 1 minus f and f squared, and there are 4 forms in general, but there's 2 that have the same outcome here. So I'll summarize that by sigma equals 0 plus or minus 1. Okay, so those are the 3 states. And so now I can write down a kind of a master equation for what's going to happen. And the next generation will be given by a sum of all the places that the particle or the bacterial concentration could have come from. So I sum over sigma equals 0 plus or minus 1. Here's a transition rate. And this is a transition rate into the state f into the particular rung of the ladder here. It could be any of these rungs of the ladder. Notice the transition rates are going to depend on where I am on the ladder. They f times 1 minus f. They might be biggest when it's a half, at least for this term here, and so forth. So I have these transition rates, and I'm going to be coming from f equals f minus sigma over n, where sigma is 0 plus or minus 1. Okay, sigma can have either sign, or it could be 0. So here's the transition rate, and then this gets multiplied by the probability that I was at a particular place in the previous generation. So here we are stepping forward in time, and what do I have? I have u of s minus sigma over n. That's the previous generation. I started at f0 way back at t equals 0. I want to know what's happening at a certain time interval in the previous generation. Let's call that t. I want to step forward in time here. So that's really all there is to a master equation. And if you stare at this and you say, well, the population is maybe 20 or reasonably sized, then a natural thing is to expand this equation. This is always true. It's just combining probabilities. So product of events that have to occur in sequence, summed over all possibilities, and so forth. So we expand in, what's the small parameter here? Sigma can't be bigger than 1, so it's 1 over n. So basically, you're going to expand in sigma over n on the right-hand side, and we will expand in delta t in the left-hand side. And you have to, of course, show that this is consistent. We'll go to second order on the right-hand side, only first order on the left-hand side. And there's subtle issues whether that's justified or not. Same kind of derivation you do for a Fokker-Planck equation. But because the rates are frequency dependent, you get something that doesn't look very much like Stokes-Einstein diffusion. And of course, it isn't. It has these subtle differences. And this is the equation that comes out. I'll let you work through the math. But you see that there's this frequency-dependent or percentage-dependent genetic diffusion constant that goes like 1 over n. And again, n has to be large for this to work. So here's the wiggles that we saw with that populist program. And you can run many realizations. And what happens, not too surprisingly, if you start off at a 50% probability at the origin, they all start like that. Then there's a probability cloud. It's like diffusion near the center of this frequency range from 0% to 100%. And then it spreads out. It sort of is a Gaussian. But there are these absorbing boundary conditions on the left and on the right. And notice what happens to the diffusion constant at the boundary. Can anybody tell me? When I'm at the bottom or the top, you can't diffuse. It goes away. You're stuck. So it's a diffusion constant that varies dramatically when you get near the boundaries. So the diffusion constant goes away when p equals 0 or p equals 1. And so once you random walk your way to 0% of capital A or 100% capital A, you get stuck. Now, it's not a simple analytic solution of this. There is for Fokker-Planck equations, not for this one. There will be one in a minute that I'll show you later. But you can solve it numerically. And that's what Kumura did back in 1955, starting with a sort of a delta function here. This is the allele frequency space represented as x. And you start with a delta function here, and you get these sort of eventually you get a delta function distribution out at concentration 0% or 100%. And this is a population of fruit flies that have been bred there are 16 of them. And they go out. Some poor graduate student had to do 19 or 20 generations of these fruit flies. And they chose some neutral mutation some BW75 variation that was easy to read off. Maybe they had black eyes or black-white coloring or something, I forget now. And then they had a wild type, but they started off with a 50%, and they could all interbreed freely. And not only did they do it for 19 generations or so with populations of 16, they did 107 of those populations, thus enabling good statistics. And it's not a perfect match, but it shouldn't be if this is only really true for a very large n. But it roughly follows what Kumura predicted: a diffusion out to the wings, fixation of all little a or all capital A. Okay. Any questions? Okay, so what about, so this is what we just talked about. Here's a summary. Finite populations, n finite, go to fixation, all little a, all capital A for long times. What is the probability of fixation starting off at a certain frequency? Well, basically any one of these, so here I have 1 out of 20 of these initial conditions being capital A and 19 out of 20 being little a. Everything's neutral. And so there's an argument that says, well, democratically speaking, since there's no selective advantage, any one of the 20 could win and take over the population. And so there'll be a 5% chance that the capital A will fix, and it's related to n. That's the idea. And then, as I said, in the vicinity of these expanding populations, like on a Petri dish, or even humans coming out of Africa, the effective population sizes are small. And so these fluctuations are big. Yes?
So I didn't completely understand the definition of the time scale. What is the small?
This thing here?
Yes.
Okay, thank you. This delta t is one of those Moran steps, which is simply allowing one cell to potentially reproduce and another one to die. The time in the test tube is playing out in parallel. Every cell is, they're not looking around and saying, okay, it's your turn, my turn, so forth. They're all doing it, it's massively parallel in the actual laboratory time. And so the Moran model tries to simulate that by doing this n times in a test tube to represent one generation. So that's the idea. Okay. And there are other ways of doing it. You can allow them to have lots and lots of gametes like the sea urchin and then just pick them out.
So,
yep.
So you have mixed delta T and tau T there? On the
yes, thank you. So yes and no. I'm including both of them. If you, and what I should have said, thank you for the question, is you do this expansion and then you repeat n times to get one generation. So what I showed originally until you corrected me was what happens for delta t, what happens in delta t time step, and there's just this 1 over n. And then I do that over and over again to build up an entire generation. That's on the left. And then I do this over and over again, adding up various variances. And the second order term in F has in front of it 1 over n squared. Then what comes in is the expectation of sigma squared, which you can check if you want to do the calculation. But then, since I do it n times, that's what knocks down this 1 over n squared when I do the second order. You have to go out to second order to get something non-trivial, and that's what gives you an 1 over n as opposed to a...
so this, when you make this
yes?
branching, this one stepwise branching, it's for the delta t.
Delta t, thank you. And then I would build up, I would have n of these to get a generation. And then this differential equation is for the generation, regarded as a short time. You had a question? Yes, please.
Does 0 stand for the space? Just like in a test tube, we have fixed amount of space?
Okay, no, it's not. We assume that these bacteria are so dilute that there's no problem of self-avoidance or crowding in that sense. There may be crowding because of nutrients, but they're not jostling each other. Sigma is simply a label for these three possible transition rates. So the one over here would be, this is just a shorthand for the formula that goes, sigma equals 1 is a shorthand for this process where you go up the ladder. Sigma equals 0 is a shorthand for these two processes which occur with this probability where you stay the same rung on the ladder in time delta t, and sigma equals -1 is going down.
This 0 in u as a function of f naught.
Okay, sorry, so I didn't understand the question. So here, oh, this 0 over here, excuse me. So this is the initial time. Thank you. So this is a function of 4 variables, but it's kind of, it's just being a little overly precise mathematically, but I should do it so that it makes sense. At time t equals 0, it could be 0. I have this fraction, and then at time t plus delta t, I have this fraction. Okay, thank you for the question. Other clarifications or questions? Okay, so we're almost there. This is a summary of what's happening, and now I'll just mention that it's not hard to put in the bias that someone mentioned. Suppose the red or the green are out reproducing each other, and here's a very simple scheme for the bias. You would say that okay, phi of f and say sigma over n. Basically, when you list these three possibilities, you say, okay, if it's going to go up the ladder, it would normally, if it was neutral, be f times 1 minus f. But I'll say, oh, those vigorously reproducing organisms that are green, they actually have an enhanced probability of being picked to reproduce, which is not f, but f times 1 plus s over 2. And then the death, I'll assume, is independent of color. So this is with a selective advantage. And then that would be sigma equals +1. You go up 1 on the ladder. If I stay the same, well, let's assume that just stays the same. You multiply by 0 anyway, most of these averages. And then down here, I would say, okay, I picked the red. Instead of probability 1 minus f, it's slightly inferior, so I'm going to give it a little lower probability than it would normally. And then I have an f there. So that would be sigma equals -1. Repeat the algebra for delta t. Repeat it n times to get a complete Moran time step. And I'll let you work that out or look at that reference if you want to look at the details. And now we get something a little more interesting. Here I've put in the question that you asked about, where what are these variables f0 and t0. So I had fraction f0 at time t0 and a fraction f at time t. And now we get the term I had before, which is genetic drift or random number fluctuations. And now I get a bias. Okay, and it turns out that this bias is sort of just what you might have expected. If you neglect this term here, and you just sort of take a look at what's going on, what you'll find is that up to noise, this fraction df dt is equal to s, the selective advantage that I just introduced here and here, times 1 minus f. Okay, I just ask you to accept that. It's plausible because now superimposed on noise, so there'll be noise here as well. Okay, and I'll just symbolize the noise by putting in f times 1 minus f over n times some stochastic function zeta of t that has to be interpreted with the Ito calculus. There's some noise. Maybe I'll just leave it this way as noise. If I leave out the noise, what's going to happen? What's the long-time solution of this equation? If df dt and s is positive, it's going to grow. It's going to saturate at 1. I invite you to check that the solution of this differential equation is f naught e to the st over 1 plus f naught e to the st minus 1. And that's basically says that if I don't have any genetic drift, if I didn't have any noise, f of t would just drift higher. Well, it would be biased towards growing exponentially and saturating, just like our introduction to the Fisher wave. But now it's not the actual population density that's growing exponentially and saturating. It's the fraction of, say, green, if green has a selective advantage. And again, it's to get some sort of intuition of what this more general Fokker-Planck equation means. It really helps to go back to that program, Populus, and make a few runs. So let's do that. I'll come up here, and I'll go to the Mendelian genetics with drift and selection. So I'll do that one. And so drift and selection. This one I'll get rid of. This one I will run. And I'm going to set this up in such a way that we have, this program was written for diploid organisms like ourselves with 2 chromosomes. So I'm going to adjust it so that we have a reasonable model. So we have a runtime of 500. And let's go to a population of 50. Let's do initial allele frequency of 5%. And let's run this thing, see what happens. OK. All right. So here we have exponential growth and saturation with noise. Okay? Say, OK, it's noise, and I just do perturbation theory and the noise and so forth. But this noise, because of the absorbing boundary conditions, can have drastic consequences. Because if before you escape this region near this perilous absorbing boundary condition, you hit it, then it's all over, even though you might have a selective advantage. And so survival of the fittest is having a very delicate interplay with survival of the luckiest. And you see, as I cycle through these, sometimes it looks like it's going to be successful. It's escaped. The absorbing boundary condition is going to be absorbed up here, and allele capital A is going to triumph. But it doesn't have to be that way. And population geneticists like to imagine that maybe over human evolution there were all these great favorable mutations like X-ray vision and wings and ESP, and how many of them died out? And we'd like to know the fraction that will make it to this final state. Any questions on the first half? Lingering questions? Yes, please.
Maybe, I don't know if you still remember, but this Verhulst term, which
yes.
was like the depth.
Yeah.
Can you explain why that's is c squared, like 3 times c squared?
Yeah, thank you. So in the Malthus-Verhulst picture, we had a population that was doing this. And why would there be a term like c squared? There's a couple answers to that. The one is that you just rewrite this in terms of an effective. It's almost just a semantical thing, but you would say that you could rewrite this as an effective growth rate that depends on the concentration of bacteria. Okay? I haven't specified what this function is yet. But there's some growth rate when the concentration is 0, which we'll call A. And it's plausible that if I make the concentration bigger and bigger, the effective linear growth rate is going to drop. And if it's anything that goes through 0, I can linearize and get an approximation that basically is what I had, which looks like this. But it could be some polynomial in C, as long as it drives the population down when the density is high. You could also say that these bacteria are, so that's one argument. The other argument, which actually has number fluctuations underlying it, is that these bacteria are wandering around in the test tube, and this is the world line of a bacterium. Okay? There's a process like this where the bacteria wander into the same point in space. So now let's imagine there's some spatial variations that are possible. And when they get to the same point in space, in a given reference volume of nutrients, there's only enough food for one organism. And so coming out of this box will be a single bacterium. We can't tell which one. And this death process will be proportional to the concentration squared. And so that's another source of this term. And in this same Feynman world line picture, you can say that cell division is represented by a bacterium that splits into two. That's births. Or if the bacterium dies of old age, that's death. And if we take into account the finite size of the bacteria, and we try to draw it in a two-dimensional space-time, this becomes the pair of pants diagram of string theory. And so forth. Other questions? All right. So we can now start, let's go back to space, all right? And let's ask what's going to happen in the laws of population genetics and so forth when space is important. I can now tell you about gene surfing. So how many of you have surfed off the coast of the Netherlands? California? California? Hawaii? Anyone? Okay, so here's a population wave, and I want to show you how genomes can surf on population waves. So this is just the population, the Fisher wave thing that we were talking about. And here in the foot, let's say it's in the foot of the Fisher wave, is a mutation. And it could be a favorable mutation, unfavorable, but let's just for now say it's neutral. Well, this is where the mutation and its offspring would have the greatest chance of taking over because the population density is low in the foot of the wave. If it had occurred back here, it would be stuck competing with 100 other organisms in its own little reference region and wouldn't have much chance because of genetic drift of the absorbing boundary conditions of taking over. But here it has a chance. And so notice this nice smooth function. It can't be smooth forever because down here we get to less than one organism. And you can't have smaller than one. And that Fisher wave stuff that's so beautiful is actually slightly inaccurate down here because of number fluctuations. The same thing that we talked about in genetics. It's supposed to go exponentially to 0. Well, it can't because when it drops below 1, it's going to be rigorously 0. So, but anyway, down here the numbers are small. And so now let's work in a co-moving frame. This is moving forward at velocity v, right? So we stay in that frame, we move along with it, we see what happens to the descendants of this mutant. And it could be that it fixes locally and takes over the population, thus providing an example of successful surfing. The wave comes by, the genome mutates locally, just the right time and place, and if it's lucky enough to escape genetic drift, it will take over. But it could be that, well, it has a chance, but the wave goes by a little too fast. It's now stuck back here, competing along the x-axis with vast numbers of the original strain. It might not die out, but it's no longer at the front of the wave. And this is an example of unsuccessful surfing. How many of you experienced unsuccessful surfing? Anyone? Okay, so it can go both ways. All right? And we wanted to find a way to study this surfing phenomena in the Petri dish. And here's just to remind you how we got started. This is a simulation. Somebody pointed out the break. These E. coli are rod-shaped. And this is the monolayer growing up. And it has lots of interesting features. Notice the tangential boundary conditions around the edge of these rods. Notice there's a defect here, what's called a disclination. And I kind of like to think that it looks like the mirror image of Africa. And if you just put in numbers for E. coli that don't have flagella, that are dividing in this fashion, and large mammals like humans that you're tracking their mitochondrial DNA. By the way, mitochondria are the descendants of what? Bacteria. Okay, so it's not crazy to use bacteria as a surrogate for the mitochondrial genomes that are used to track these range expansions. And the bottom line is what I said earlier, is that in 500 generations, you go 10,000 kilometers here and you go a half a centimeter here. So let me go back to that experiment that I started this discussion with. Here are two E. coli, schematics of two E. coli genomes. This is the main chromosome, main chromosome. Those chromosomes were identical, they were clonal. And then there was a plasmid, a second chromosome. And these plasmids were also nearly identical. They were quite small compared to the main chromosome. And on the plasmid, which were identical almost all respects, there was one change. And it was the change in a constitutively expressed, that means a gene that's on all the time, in the color of the fluorescent protein. So this was, say, yellow, and this was cyan. And in fact, it only took one amino acid change to shift the color. So they were as identical as we could make them. And we mixed them up, and somebody asked what was the protocol, and the protocol was inoculating down a few microliters in a pipette. The carrier fluid evaporated away, and then under high magnification, after the evaporation, this is what it looked like. 4 days later, this scale is about a centimeter, we got this rather boring-looking white coin-shaped colony growing on the Petri dish. How many of you have done this experiment where you shockingly discover what's inside your mouth by putting a little wire, putting it on the Petri dish, looking at it a couple days later, and it's all furry and horrible, and that's why you should brush your teeth? Okay, so this is the same experiment, basically. But we had these two colors in 50-50 proportions, and under fluorescent excitation, it looks kind of interesting. Here's a more global view. So this region inside the white circle we call the homeland. There's lots of stuff going on just outside the homeland as this great migration proceeds outward in a radial fashion. But eventually, genetic drift at each point along the perimeter is going to cause fixation of green or red. And when it's sort of yellowish in here, it's because they're still fighting it out. They're still in the middle of this 0% to 100% gap that we've been talking about. And since you're starting off at 50%, half of them end up red and half of them end up green, and then you're stuck. But different outcomes happen, different histories as you go around here. And so my colleague, Stephen Jay Gould, looked like to speculate what could happen if you replayed the tape of life. We can do it with these organisms, at least in the context of these simple homeland inoculations. And of course, the details are different. Here we have these genetic boundaries, but there are universal aspects, we think, of what happens with these range expansions, as I will discuss. And it doesn't have to be a 50-50 mixture. In this illustration of survival of the luckiest, you can see that this is a 95% mixture. Most of the red around the perimeter of the homeland, you can see their fraction you can estimate it by looking at the area in the interior, which is red. But of that 5%, some of them started off, but then got squeezed out. Here it got started off, and then it maybe survived. This one definitely survived. And this is an example of gene surfing. Okay? And if you blow this up, you can see, you can almost trace back and find the bacterial Eve that determine the time and place that Eve gave rise to this progeny that came out. Yes, please.
Why is there so much contrast in the homeland here?
By contrast, you mean you see red and green and...
Exactly. So the red is really focused already.
Yeah, so that might just have to do with the way we set up the microscope and so forth. What we think happens is that, like in this previous experiment, we end up, half of these are red and half of these are green. This will create a little red colony. This one might create a little green colony. Then they collide. And probably we weren't good enough on the microscope to get them equally balanced in the sense of your interesting question. So I think that's my best guess as to what happened. And notice that because these stop dividing, we have a record of the entire history of the range expansion. If this is the present day, we could make measurements around the rim and maybe infer the size of the homeland. And we can see whether we can do that accurately by trying out our theory on a repeatable experiment in a Petri dish. That was our goal. Here's another experiment which I did one evening with a razor blade that I sterilized over a Bunsen burner, and then I touched the razor blade to a liquid pan that had the two different bacterial species in it, touched it to the Petri dish then, and so we had a range expansion up and a range expansion down. Okay, and so we can think of, if I look at I look along this black line, what's happening is exactly like that serial dilution experiment or that chemostat experiment, because if I go from one generation to the next, what's happening is a variation due to number fluctuations of the fraction of red and green sitting inside each generation. So these are the actively dividing patches in the front moving along this black line. So rather than, it's as if the range expansion does serial dilution for you. The proportions will be changing, and in some places red will win off this linear inoculation. In some places green will win. If you are a dust mote sitting on the Petri dish looking at this tsunami of bacteria coming at you, at sort of your eyes were down there in the Petri dish, you'd see this mixture of things coming initially, but then it would segregate into red sectors and green sectors, as you see here. Okay? And so, as I said, these generations map onto this population genetics thing. And if there were a selective advantage, that equation, that Langevin type equation thing that I had earlier for selection with drift, here's selection, here's drift, would apply. And as we saw with the populus program, on average, maybe you might go up and saturate, but there'd be this noise, and that noise could sometimes be deadly, causing even favorable mutations to die out. That's an issue for tumors. We may not get there today, but if you have a tumor, I don't actually have a tumor, fortunately, but I do have this ball-bearing array. And think of this as a growing mass of cells in the human body that has gotten a little out of control. It's not really cancerous. It's a benign tumor. It's growing slowly. But what people think about cancer, I'm still not an expert, is that it requires 4 or 5 or 7 more mutations for the cells in this slowly growing tumor to become malignant. They would have to acquire the ability to throw off all restraint and divide like they were bacteria again, or yeast or something, rather than the controlled cell growth, the carefully controlled cell growth that characterizes a healthy human body. That would be one mutation. Another mutation might recruit blood vessels to supply nutrients to the interior of the growing bolus of cells, because there'll be nutrient shielding, just like you can't grow in the middle of these two-dimensional colonies. And now this green represents one of those mutations. There might have to be 5 or 6 of them. And the question we really care about is whether this green mutation on the surface of a slowly growing tumor will surf. And as it grows, take over. Eventually you would have all green on the surface. And then well, after that happens, there'd be another one, and then 7 other mutations later. If you were unlucky, you'd have a full-blown malignant rather than benign tumor. So it's an analogous problem in 3 dimensions. So I'll pass this around if people are, it's not really a tumor, don't worry. So that's the kind of question. Can that little green splotch overcome the genetic drift to spread all around the surface of that sphere? Or it could be a circle or whatever. Okay, so how do we deal with these spatial problems? I'll just tell you briefly because I want to get to some of the other stuff here. And this is a stepping stone model. And you can imagine that each point on this surface, like this is in white light after 10 hours with one of those razor blade inoculations, each black arrow is doing this, but the black arrows talk to each other because the jostling cells at the front are pushing each other left and right, and so there can be effectively some kind of spatial communication or spatial diffusion. Not genetic diffusion, but spatial diffusion between neighboring arrows, let's say. And the stepping stone model is nothing more than a bunch of little population genetics reactors that allow also some exchange of cells from one to the next. And if I sit in a local reference frame moving with this outgoing population wave, I have a one-dimensional stepping stone model going along this frontier. And so again, we could have a fraction of red. Because it's red instead of green this time. And the ith stepping stone, so this would be I equals 1, 2, and 3, will have a certain fraction. There's a genetic diffusion constant that we've talked about. Some conventions have a factor of 2 there, some don't. Don't worry about that. And this m is another diffusion phenomenon. It's a spatial diffusion constant. And if I just put in mutations, basically all of population genetics is summarized in this master equation, at least in this one-dimensional context. You can also do it in 2 dimensions. This is the genetic drift we talked about in the first half. This is just a discrete version of the spatial diffusion operator. And if you wanted to put in something involving mutations, I'll just write down the equation, and you can sort of get a sense of what's happening. There'd be spatial diffusion, say in higher dimensions. This is the fraction of green. In one of these models, say on the surface of that sphere, there would be S times F times 1 minus F. So that's the selective advantage that we've talked about. If I want mutations, then there would be, say, a mutation rate out of the state F, say from green to red, that goes like that, and then maybe another mutation rate nu that goes like 1 minus F. And then finally, there'd be this genetic drift term, which would be the square root of F times 1 minus F, either n or 2n, times some noise that would depend on space and time. Okay, so this is a kind of master equation for all of spatial population genetics, which, yes? Sorry? Oh, thank you. So one is negative and one is positive, sorry. So to answer your question, if green mutates to red at a rate mu, that would be summarized by this negative contribution. And if red mutates into green at rate proportional to the amount of red that's already there, that namely 1 minus F, that would be this term. So this is mutation. This is, as we've talked about, selection. This is Darwin. Darwin didn't know about genetic drift. Knew a lot, but didn't know that. And this is diffusion. And this is noise, but population geneticists call it genetic drift. Random drift. It's not, there's no bias here. It's just going back and forth as we discussed. Okay? And so just extend this, this Laplacian in one dimension discrete model is equivalent to this term here, it turns out. And everything in say, a linear range expansion is going to be described, we think, by this model. And It turns out, amazingly, because there's this nonlinear noise, it's exactly soluble. And you can compute something called the heterozygosity correlation function, which is the probability if along this razor blade you measure one color here, you go a certain distance away, probability the color has changed. And that depends on distance. If it were an Ising model, and green were up spins and red were down spins, we'd be looking at some non-equilibrium kinetic Ising model at a spin-spin correlation function at a fixed time. And this can be calculated, and at time t equals 0, it's just flat. If there's random initial conditions along the frontier, there's equal probability of getting red or green, and this ends up being a half. And you can do simulations. This is an experiment off a razor blade, and you get pretty good agreement. But there's a subtlety that I haven't focused on, but I'm now going to tell you about. This is the razor blade inoculation, and this is the heterozygosity and so forth. Most of these range expansions are on circles in a terrestrial environment. If a foreign organism lands on a coastline, it would be semicircles as it invaded into the virgin territory. And these radial growth problems are slightly different because what happens is you get random walks, but they're radial random walks. Up here, the random walks are going around. They go like the square root of time, these genetic boundaries. They collide into each other and pinching off and so forth. And that happens for a while over here and over here for E. coli and Pseudomonas aeruginosa. But eventually, the perimeters, just like the inflating universe, are getting further and further apart because you're going out linearly in time in a radial direction. Just like my arms are getting further and further apart. The tips of my fingers are further apart than my elbows. And that's a linear inflationary separation, which eventually wins out over the square root of time annihilation of these random walks, like up here, up here. And if you work it out, you can show that eventually the number of sectors, up here we think that either green or red would win at long times, going up or going down. But on with these radial expansions, eventually inflation sets in and the number of sectors stops changing. And you can show that the number of sectors in this simple random walk model goes like the square root of the initial radius of the homeland times the velocity times this divided by the square root of twice this a diffusion constant that basically the spatial diffusion constant describes the wiggling of these boundaries. The hard thing was getting pi over 2. The rest of it is sort of back of the envelope estimate. But notice what you can do. You can go around and count the sectors once you reach this inflationary epoch. That you can get experimentally. The velocity you can measure out at the rim in the present day. And you can measure the wiggling of these lines out at the rim in the present day, invert this formula, and determine the radius of the homeland. And you can see, by repeating this experiment 40 times, whether your theory is any good. So that's the idea. And there are error bars. The theory says error bars that you can also check. So that's the idea. And here's showing you that we can do replicates. We can calculate heterozygosity correlation functions. In a radial context, you go to this universal shape at large times. I won't spend a lot of time on this because I want to flip and tell you about some very recent work with Max Leventovich on inspired, hopefully, maybe a little bit of insight into some small corner of issues involving cancerous tissue. But this is just showing you that we can do experiments on, this is yeast. So this strange shape is just, it's budding yeast. It's spherical organism. It has a nucleus. It's a eukaryote. And it buds off smaller daughter cells. The blue one can't make leucine, but overproduces tryptophan, which is great news for the yellow one, because although it overproduces leucine, it can't make its own tryptophan, and so they have to play together. And so the question is, will this pair exhibit genetic demixing as neutral strains might? And I won't go through the math. You can work out what's going to happen with a generalized version of this model. Notice the selective advantage term has changed a little bit. But I will tell you that if you work out, there's a phase diagram, and only for a limited region of growth parameters, alpha and beta, do you get this mixing. So if I go back here, maybe I'll do it on the next. Here we go. So the way you do this is you say, aha, now there's a frequency-dependent selective advantage that comes in front of f times 1 minus f, which we've seen before. And that comes from these different growth rates of red and green. And so G is a background growth rate for both of them. But beta is a coefficient that tells you how helpful it is for the red to have a green nearby, because this is the concentration of green. And the growth rate goes up if beta is positive. This alpha tells you how fortunate it is to be, for a green, to be near a red with concentration F. And that coefficient is alpha. And as you work it out, there's this kind of inverted W-shaped potential that describes what's going on. And you can work it out in space, as Kirill Korolev did in a paper we wrote together. And there's a phase diagram. It's a function of alpha and beta. In here, there's mutualism. In here, say, blue or green wins. Over here, there's the genetic demixing. Like that razor blade inoculation took place right here at the origin. And that behavior continues along what seems to be a kind of line of first-order phase transitions. Here, the other color wins. And this is an experiment. The experiment is really quite beautiful. It was done by Melanie Mueller. So I think I can say it's beautiful, as she did it with help of Andrew Murray. And what was done here on the left was to mix these guys up in the homeland and to have a plate, an agar plate, which had some nutrients, but absolutely no leucine, absolutely no tryptophan. So to survive at all, they had to stay mixed. So he managed to suppress this very powerful force of genetic mixing, genetic demixing. However, if you add tryptophan and leucine, so they don't need to cooperate, they can get as much of it they want from the plate, they start genetically demixing over here. And somewhere between here is a phase transition. And that's what we're studying now. Any questions on this part before I switch gears a little bit and talk about mutations? OK, so let me go over to another talk. This is actually similar to a talk I gave in Paris last week at a conference on physicists meet cancer and tissue growth, or some slightly strange name like that. So I passed around this tumor. I noticed a few of the cells have migrated, actually, as it went around. That's interesting. The spatial diffusion is happening on the surface of a sphere. Here's all that I'm not an expert. This is what I think is useful to know about avascular tumor growth. Avascular means that you haven't got you haven't gotten capillaries and arteries in there to supply nutrients. There's a thin growing region on the outside of the sphere, just like we had a thin growing region on our bacterial colonies at the edge. There's a region of quiescence, though they stopped growing like the bacteria. Never got this severe in the interior of these two-dimensional colonies, probably because they could get nutrients from the third dimension. But here, you can't get nutrients from the fourth dimension. You're already in 3. And so there's a necrotic core where there can sometimes be volume loss. And this is a really cool experiment that Max Leventovich and John Kashwanis did, where they looked at hemispherical colonies, not of actually tumor cells, although we hope people will do this experiment, but yeast cells. And basically, when they get big enough, green, which indicates active cell division, only occurs on the rim of the hemisphere. Not just the rim, but the whole hemispherical surface. Red means this region here of quiescence, non-division. There's still proteins produced that are red, but you only get the green ones on the outside. And there's a nutrient screening length showing dramatically, in this case, that you only get growth a certain distance in toward the center. So it's very much like a three-dimensional version of what we've been talking about with these microbial colonies. And as I said, cancer often results from genetic changes in somatic, that means not the germline, but the cells in the body, they stop cooperating. It's amazing what the cells in the body do. I mean, they commit suicide when they need to, they stop growing, they're very polite, well-mannered, and occasionally we get mutations that cause growth that can lead to these abnormal forms and so forth. And as I mentioned, you might need a number of driver mutations to take over at the surface, and then eventually you'd get something at the surface where they might presumably have room to grow, which would have enough of these mutations, 5 to 7, which can lead to serious problems. Malignant neoplasms, I think, is the fancy name for it. And I just want to ask, what is the probability of surfing on the surface of a sphere, of a spherical cell cluster, and could we use some of these ideas? And this is an existence proof that you can study these things in vitro. In the laboratory, there are cell lines that came from breast cancer, and most of the time they're reasonably well behaved and they just slowly grow spherically, but then you can activate them and enhance their growth rate. And so that's an example. This is Max, who's just finishing up his thesis and did most of this work, and we had lots of help, as usual, from these wonderful collaborators. So that's what I want to spend the last 20 minutes on. Any questions at this point? Okay, so here is more pedestrian but still quite beautiful experiments in Petri dishes. This is not a cancer cell line. This is Penicillus dendriformis, which means the praiseworthy bacteria that can form dendrites. This doesn't form dendrites under these conditions, but it does have mutations. Look at this mutation here. It just has a slightly different coloration, so you can see it right away. This is the basic experiment of microbiology. People have been doing this for 150 years or so. And if you see something like that, you might these days scoop out this little piece here, go sequence it, and see what's different from the host strain. This is a mutation. This is wild type. The mutation is taking over in some interesting way. Okay. And we went through a little bit about this razor blade stuff. I just want to tell you briefly that this is yeast, but if you do a razor blade inoculation of yeast, you can use the growth velocity of the red versus the green strain as a kind of proxy for the selective advantage. Survival of the fastest. And so we could put in the formula for the Fisher wave growth velocity of the background green strain, and then compared to that velocity, we'll just say that the red strain, the mutant strain, that's what M means, is V times quantity 1 plus S. So S is just like the selective advantage we were talking about. And it turns out, if you just measure this angle at the end of the day, you get the selective advantage. And that calculation is high school trigonometry. You just say, oh, well, calling out this little sector here that ultimately has a slightly bowed frontier, I go along this distance, and basically this point, this distance had better match this distance. They better arrive there at the same time, these two population waves, the one that went along the inside of this dashed line and the one that went on a straight line shot. The one that went on a straight line shot is going a distance t times v, but that has to equal this distance t times the velocity of the mutant strain in the middle times the cosine of phi over 2, trigonometry. And if you solve for what the angle means in terms of the selective advantage, this is the formula. The angle is twice the arc cosine of 1 over 1 plus s, or for small s, it's often the case, it goes like the square root of s. So if you're good enough to measure an angle to 1%, and everything else worked beautifully, this has not been done, but might be able to in the future, 1% in phi would give you 1 part in 10 to the 4 in selective advantage. So it might be a useful way to read off these selective advantages. And another way to say it is there's a perpendicular velocity here of the front at the frontier, And you can get that from trigonometry also. And now, what is the fixation time? The fixation time, if this domain on the razor blade has size L, it's going to be L divided by this perpendicular velocity. So it's L divided by the square root of S, turns out. Okay, so there could be, we want to know, in the case of a spherical tumor, this is a linear problem in 2 dimensions, but in the case of a spherical tumor, we want to know how long will it take for these green cells to sweep over the surface. And if it were flat, then that time would be proportional to the linear dimension. It might be the perimeter if it were a circle, divided by square root of S. But this is flat. It's not a circle. But you can get the selective advantage. It's about 13%. OK, what's going to happen for a circle? Then we will do a sphere. Okay? Circle is a little easier to do mathematically. So let's take a little colony here. This is a simulation, not an experiment, but I'll show you experiments in a minute. And at 12 o'clock, we'll put a very small piece of mutant that grows faster. And what Kirill Korolev did, part of his thesis, was to solve some reaction diffusion equations. Mutualism is described by these epsilons, if you want to put it in. He usually didn't. Then there's diffusion, spatial diffusion, and stuff like that. And you can figure out what's going to happen. Anybody have any suggestions about what might happen if I run these simulations forward in time or did an experiment? Let's suppose it's a favorable mutation, so S is positive. Let's take a look. So here it is, this gray. That's the mutant. It grows. It starts to envelop its host. Eventually, makes a beautiful heart. It's discovered on Valentine's Day, actually. OK. What's the shape of this heart? Well, this is not high school trigonometry. This is a little bit of differential geometry. So you basically say, OK, there's some curve. I'll go to radial coordinates. This is the angle. This is the radius as a function of phi. And this distance here had better of the mutant had better match this radial distance of the wild type. And if you work that through, I invite you to do it, you discover that in terms of the mutant radial velocity and the wild type radial velocity, the angle is given by the logarithm of this radius. And if you put in Vm equals 1 plus s times v, discover it goes like the square root of s again, but it's multiplying a logarithm. And so now we can ask, what is the fixation time? How long is it going to take to make this heart? But it's curved now, so it's a more complicated calculation. And you can get it by just setting phi equals pi, otherwise known as 180 degrees. Okay? Now, the fixation time, which is related to a fixation radius as the heart goes around, is the radius of the homeland times e to the pi over square root of 2s. So now, for small s, this is exponentially sensitive to the selective advantage. Instead of 1 over square root of s, it's e to the 1 over square root of s. And that's probably worth knowing. Why would you want to know that? Well, you'd like to know if this mutant green is somehow expanding in a spherical analog of what I've just described. If it's going to take a really long time to envelop, maybe another mutation will come up at the same time and get in the way or change. It might even be a deleterious mutation. And so that's called clonal interference. And to decide these issues of clonal interference, it's important to know the fixation time. And here we have a simple formula for it in 2 dimensions. Now it turns out this same, well, let me summarize. Here's the argument. This is a famous curve in mathematics called a logarithmic spiral. It was discovered by Jacob Bernoulli. It occurs occasionally in physics, but a lot of times in biology. It describes the shape of a trajectory of an insect that's attracted to the flame of a candle. It goes around and around and around, and it comes in to a fiery death on a logarithmic spiral, related to the fact that it has eyes a little bit off center. And why would it do such a stupid thing as to kill itself, or you know, a mosquito trap at a party where you have some UV light or something that could, a bug zapper? probably because that algorithm, if the source of light were the moon or the sun and was effectively infinitely far away, that would enable it to fly in a straight line. But it evolved to fly in a straight line, but now we nasty humans have point light sources which cause the spiral death of these bug zapping insects. So that's one application. It's also the shape, for other reasons, of the Nautilus shell. And fossil ammonites that go back 70 million years also have this shape. And I hope I've convinced you that sectors in microorganisms also described by logarithmic spirals. Jacob Bernoulli was so pleased with his discovery, he said, I want a logarithmic spiral inscribed on my tombstone, on my grave. So this is the brass inscription. It says something about what a great guy he was. But sadly, the engraver inscribed an Archimedean spiral with equal spacings instead of a logarithmic spiral. So he got stuck with the wrong curve that he did not invent, that Archimedes invented. Not nearly as cool as the logarithmic spiral, I think. Okay, in fact, Kirill, who's very much an excellent theorist, went in the lab and tested these things. And Pseudomonas aeruginosa, here's a curve. If you plot this curve, log R versus phi, selective advantage 8%. This is Melanie Muller's data on a slightly less dramatically curved, but still it takes a while for these sectors to get established, but eventually they too form logarithmic spirals. And significantly, perhaps for these tumors, this argument works in 3 dimensions. So this is work by Martin Nowak and collaborators, and they just went through the same simple math on a sphere. And so this is like a continuum model of a mutant of a background slowly growing tumor. This is a driver mutation. This is like those green ball bearings, and the driver mutation is taking over, and you go through the argument, and the fixation time again goes like e to the pi over square root of 2s. Now this leaves out clonal interference, as they point out. As this blue starts to take over from the gray, you could have yellow, red, and green, different mutations, all getting in the way and interfering. And that's important to study. That I'm going to neglect. But there is this interesting logarithmic spiral in 3D as well. What's missing from this picture? Can anybody in this room tell me what's left out with these smooth curves? Noise. Genetic drift. Genetic drift is left out. And the crucial thing that's missing is that tumors like this and these circular colonies of microorganisms are bumpy. They're composed of discrete cells. And in fact, that's shown here. This is one of these. It's not a razor blade inoculation. My colleague Wolfram Möbius, who works in my group, basically soaks some filter paper in bacteria of 3 different colors and let the range expansion go on. And these wiggly lines, they're a reflection, as we've seen, of the discreteness of the cells at the frontier. Einstein used a similar argument when he was studying Brownian motion and said, you know, that's why we know that the water is discrete before people ever measured it, because the pollen grain is jerking around. And if the water molecules were 3 orders of magnitude smaller, they wouldn't be moving, because all the different impulses on the pollen grain would average out. Here too, we know that this is made of discrete entities because of these wiggles. And in fact, that wall diffusion constant, if it's a monolayer and only grows right at the surface, is given by the cell size squared divided by a generation time, just what we talked about in the first half. And if the cell size were 0, there wouldn't be any wiggling. So you can read off on a centimeter or millimeters size scale of this filter paper the quantization of the cells which are occurring 10,000 times smaller on the micron scale. And it's left out of that calculation. The fixation time is right once you escape genetic drift. So we have a spatial problem of genetic drift here, and we need to see what's going to happen. I don't have time to tell you, exactly what goes on. I'll just give you a flavor. What we did was we had ball bearings or ping pong balls or something on a computer. We could grow them in a planar way, analogous to a razor blade inoculation, or we could grow them in this sort of disorderly mass that's a lot like dense random packing, but now it's dense random packing with population genetics superimposed. And we would put, say, one mutant cell at the frontier and ask, what will happen? And we will give it a little bit of selective advantage, and we'll ask what's going to happen. So we're still sorting out what happens on the surface of a sphere. We think we know what's going to happen, but only roughly. We do know what's, and I'll give you the simulation that shows it. So this is the logarithmic spiral that you saw, and this is one of Max's lovely simulations. This is successful gene surfing on the surface of a tumor by a green driver mutation. But it didn't have to survive. We don't know what's going to happen a priori. And so what we did was what we asked, the most simple question you could ask, which I alluded to briefly in our discussion of conventional population genetics in a zero-dimensional test tube. We could ask, what is the survival probability of a single mutant at a frontier? And this is a problem that we can at least solve with a fair amount of accuracy in 2 dimensions. We think we know how to do it in 3. We're not sure, but we think we can do it. So here's what's happening. In a 2-dimensional world, I have a little opening angle here at the surface of the homeland in a radial inoculation. And here I have some sort of potential driver mutation. Might have a selective advantage, might not. But those random walks, those radial random walks, have pinched it off. And sadly for the tumor, but fortunately for the human, it gets encased and no longer can reproduce. Now this survival probability, which is what we're trying to say something about, depends on a lot of things. It could depend on the selective advantage, it could depend on the radius of the homeland, will depend on the size of the cell, A, depend on the generation time, on this little angle here of the initial state, on the velocity, on time. Fortunately, in the long time limit, it's not a function of 7 variables. Those of you who do experiments might be glad to hear that. And in fact, we can show that it collapses down in the long time limit only to a function of 2 variables. And for tumors, you can make a reasonable approximation, which is that the velocity of the range expansion times the generation time is the cell size. That says it grows 1 cell width like an onion at a time. And then another useful thing to say is, well, what we really care about is what happens if the mutation rates are low, if there's a single mutant at the perimeter, in which case this angle up to geometrical factors is just A over R0. So if you put that in, this becomes a irreducibly complex, but only 2 variable function of the square root of A over R0, and then this dimensionless selective advantage, which we call kappa. It's the selective advantage that comes from the velocity or the cell growth rate, which is a small number, multiplied by the square root of the radius of the tumor when it first gets the mutation divided by the cell size. This is a large number. Maybe it's 20 or 30. And so small times large, key parameter. It could be big or small. And in terms of this parameter, if it's large, there's a fairly big selective advantage. And what do we have here? We have a noisy logarithmic spiral. Here we was trying to be a wannabe logarithmic spiral that got squeezed out. And here we have, for kappa equals 0.05, a mutation whose fate is unknown at this time, at this point in the simulation. And at least in 2 dimensions, this noisy logarithmic spiral picture, or it can be described by a Langevin equation. It doesn't even have these weird f times 1 minus f's in it. You just look at the stochastic behavior of this phi of t, this is the selective advantage, this is the deterministic part, and this is noise in the usual Langevin sense. This delta has a weak temperature time dependence, and it's exactly soluble. And you can compute this survival probability. You can get the function of both variables. I should finish up. Notice there's a finite survival probability even at s equals 0. Why? Because of inflation. If there's a mutant on the surface and the surface is growing, it and its descendants are inflating away from the dangerous encapsulation by the wild-type neighbors. And that gives it a little enhanced survival probability. And if you like math, this is our formula. But it actually seems to work pretty well. And I'll close by saying that we can do simulations on the surface of a three-dimensional sphere, like this one, sort of like the surface of those ball bearings. And sadly, for the tumor, but not for the human, it got engulfed and died out. That happens some fraction of the time. The other fraction is this one that survived. The tumor is getting bigger. It's just being rescaled. That's why the spheres are getting smaller, so you can all see it in the same volume area domain on the slide. But the tumor is getting bigger, and it has this logarithmic spiral cap. And in the 3 dimensions, we can calculate the survival probability as a function of this dimensionless time. And here, too, there is a finite survival probability at s equals 0. And so neutral mutations they're sometimes called hitchhiker mutations that will hitchhike onto the passenger mutation and sometimes mess up its selective advantage just by giving some sort of genetic overhead. So that's the story. I'll end by just saying if the mutation rate gets higher, you can have lots of competing logarithmic spirals that are born at various times. This is a mutation to simply one driver mutation, one type, but really there should be many. We can solve that in 2 dimensions. We're now trying to solve it in 3. And with that, I think I will close and thank you for your attention. And I'm happy to answer additional questions if people have any. Maybe you're tired and you want to go to the bar. Yes?
Just to recap from where we started, could you also make maybe then similar images where you look at the genetic conservation of genes in the travel of the humans around the world?
Well, you could. I think we've only looked at the simplest possibility of 2 different alleles, 2 different variations. What actually happens in humans, since we have a genome that's a billion or 2 base pairs long, is that there's lots of things going on in parallel. You can think of them as different colors. And to really do the correct inference for humans, it's much more subtle than you might have thought. In fact, at least you can get a sense of how to avoid some potential traps. I don't think I'm at the point where I could reliably say anything about humans, but here's a potential trap. This is this gene surfing. So if this were humans, and there actually is Nature science papers that make such claims for humans by looking at spatial population genetics, they may have been retracted by now. You saw this and say, oh, what's going on with this red? Maybe that's, for humans, that would be like the gene for an opposable thumb or speech. Because look, the red's taking over, and there must have been some amazing thing that happened right here. In fact, in this case, that's not true. The red, it hasn't changed from its ancestral form, and it's virtually identical to green. You can do sequencing or various tests. And it's simply survival of the luckiest. There's 2 random walks, one on the left, one on the right, and they just didn't happen to meet on this time scale. And now, because of inflation, They'll almost never meet. But it's not because of some great favorable mutation that you can attribute to humans. That can happen too. Then you'll get a noisy logarithmic spiral, and if you can show that it's logarithmic, then you'll know something. So I will certainly say that we can't yet say anything directly about humans, but at least we can confront simpler questions, like sort of like the hydrogen atom in physics isn't the same as a really complicated thing like uranium. But understanding hydrogen is a first step towards understanding the rest of the periodic table, and we hope we've made a small contribution in that direction. So that would be one answer. If there are no further questions, let me wish you all the best of luck in your scientific careers, and thank you for all your questions, and good luck to you. All right.